This is a special presentation by Zorana Repac. Since the beginning of our IMZRCB conference, we've been honored to have ongoing cooperations with the Journal of Zoological and Botanical Gardens, which was newly established around the time of the conference. So I'm happy to introduce you to their journal relations specialist, Zorana Repac, who will be making a presentation on the open access journal that covers areas at the intersection of fauna and flora conservation. So, Zorana, the floor is yours.
Okay, hello everyone. I'm truly glad to be able to participate in this year's conference. Here, I'm looking forward to hear all about the amazing research that is done in ex situ conservation and present our journal. Thank you, Ido, for introducing me and introducing our journal, so to say. And I will just inform you about the journal itself. So this is international, peer-reviewed, and open access journal on the intersection of fauna and flora conservation. It's published quarterly online by MDPI, and the journal was established back in February 2020. So this is a young journal, and we've been working hard to develop it, and you will see our results a bit later. But I wanted to tell you that we publish reviews, original research papers, as well as short communications and opinion papers. We don't have any limits in the terms of length of the paper, the figures, the colors, and so on. So the journal draws together expertise from multiple disciplines. So we are trying to explore and share your research regarding various questions concerning management, conservation, and sustainability of biodiversity, and human nature heritage issues as well. The journal is indexed in DOAJ as well as Scopus and Emerging Sources Citation Index. And we are also affiliated with the World Association of Zoo and Aquariums and this conference, as Ido mentioned. So the members from the World Association Zoos and Aquariums members will receive discount when they submit the papers, as well as you all. About the journal, so as I mentioned, it is really young journal, but we managed to receive the first impact factor of 2.1 last year, and we are Q2 in biodiversity conservation as a category rank in JCR. We also received first site score, it is 3.0, and in Scopus and site score, we are Q2 in agricultural and biological sciences, and also veterinary. The most cited paper last year, you can see here, it has 4 citations already, and the most viewed paper last year was written by Paul here. It was the most viewed paper last year in the zoological topic, so to say. Regarding our publication statistics, as you can see here, since 2022, our submissions increased more than 80%. The publication rate is steady, so that is what we've been working on. So each year we are trying to publish the similar number of papers, if not a bit higher, and we are trying to attract high-quality research paper, of course. What is, so to say, famous about us is that we process the papers really fast. So if you submit your paper, so this is the median processing time of the art, every paper. So it is around 52 days, meaning when you submit a paper until you publish it, the median value is 52 days. So for some other journals, it may take 6 months or 9 months or even less of that, but we are famous about the rapid publication, but not interfering with the quality and the editorial procedures. Our editor-in-chief is Dr. Kevin Tianfalione from University of Lille in France. He's with us for 3 years now, and we have 27 editorial board members who are external editors who will help us decide whether the papers should be accepted or rejected, of course. And also we have 8 topical advisory panel members who will advise us on the journal development and promoting the journal, attracting submissions, and so on. We listed here the most active editorial board members in the past year. So if any one of you would like to join to our editorial board you're of course free to speak with me later. We also have several special issues open. The goal of the special issues is to promote the papers on certain topic, to highlight it, because from our experience, the papers published in the special issues are really highly visible than the regular papers. Regarding the online readership, in the last year, it was 100% increase, so almost 400,000 views, and also downloads was the pretty much same throughout last year. It was around 7,500. How can you join us? You can review papers for us if you have your PhD. You can lead special issue on the topic of your interest, and you may join our editorial board. What we offer to you is we provide various layouts, English editing, administrative assistance, everything we can do from our side. As I mentioned, the rapid publication. Also, if you review for us, we provide discount vouchers which you can combine, and then you can publish for free, of course. And for this year, as Ido mentioned, for all participants, we grant free papers. So thank you for your attention. If you have any questions, feel free to reach out later during the days. Thank you.
Hello. Oh, thank you very much, Zorana. I just wanted to say really quickly that there are flyers from JZBG back here on the table, so if you are interested, please pick one up. Thank you.
Good. Now we are starting with the more specialized sessions. We're starting with the session 1, which is called Evolutionary Diversity. And our first speaker is Professor Dr. Irina Ruf from the Senckenberg Forschungsinstitut and Naturmuseum in Frankfurt, Germany. Irina Ruf is the head of the section of mammalogy at the Senckenberg Research Institute and Natural History Museum in Frankfurt, like I said. And her research comprises the evolution, comparative anatomy, ontogeny, and functional morphology of the cranium in extant and extinct mammals and their ancestors. And the presentation is called, or is about, the evolution and functional morphology of the mammalian inner ear. The stage is yours.
Thank you. Thank you very much for inviting me here. So I would like to show you today some results of my own research, or my research group, about the inner ear of mammals. Does not work here? Okay. So the ear of mammals is highly sophisticated and highly derived in several respects. For instance, the pinna of the outer ear the 3 auditory ossicles instead of 1, like in other terrestrial vertebrates, of the middle ear, and also the cochlea of the inner ear. And the inner ear has 2 functions. So first of all, the upper part, the vestibular system, is housing the sense of balance, especially the 3 semicircular canals I will show you later. And the second part of the talk is a kind of proxy for locomotory adaptation, or can be used as a proxy. And then the cochlea, which is unique to mammals, the coiled cochlear duct or canal, that houses a sense of hearing. I will also show you some examples of that. So based on these modifications of the ear, the auditory performance in mammals is different from other vertebrates in that they have a wider range of frequencies they can hear, and also higher frequencies than all other vertebrates or terrestrial vertebrates. And actually, coming to evolution of the system, it's well known from the fossil records, the transition from the auditory ossicles as part of the lower jaw, and how they detach, become very tiny, and then become part of the middle ear. So there's a transition from substrate sound transmission to airborne sound transmission. This is very well known from the fossil record. Less is known about the evolution of the inner ear structures. And how can we study these very tiny organs? It's only a few millimeters in length. So based on modern imaging techniques like micro-CT, we can more or less non-invasively study these organs. So we do micro-CT scans, and as the inner ear is housed in a very dense bone, which is also quite well preserved in the fossil records in many species, we can virtually extract the hollow space of the soft tissue structures. And this hollow space, the bony labyrinth of the inner ear, actually represents more or less one-to-one shape and size of the soft tissue structures, and can be used as a proxy. And by doing so, we can study clean skulls of extant species from museum collections, but also fossils. When we take a look at the extant major groups of mammals, the therian mammals, marsupials, and placentals, how do their inner ears look like? So they all have a cochlea that has at least 1 full turn. So this is a definition of theria. And there are some species like guinea pigs who have up to almost 5 turns. Instead, monotremes, the platypus and the echidnas, look a bit different. Their cochlear canal has only a half turn in the platypus and 270 degrees in the echidnas, and there are also some other difference in their anatomy I cannot deal with today due to the time constraint. So I will just go for the shape of the cochlea. And this is also the first topic of the talk. It's the evolution. Nowadays, we have a good glimpse into the evolution of mammals. Since the Triassic, around 250 million years ago, there's a whole bunch of species described. Here you see a phylogeny with the modern representatives. I'm sorry, this was a wrong button. Here up the monotremes and the therians, and as you can see, there's many species and major groups in between in the phylogeny. And what we already know since long time is that the ancestors of these mammals, also the mammals in the so-called Mesozoic, the Mesozoic mammals, they don't have any kind of cochlear canal or cochlea. It's simply this small, tiny sac on the inner ear, on the bony labyrinth, where the sense of hearing is located. Then, going up the tree and going forward in time, already basal Mesozoic mammals like the Morganucodon had an elongation of these structures, so it's a beginning of the cochlear evolution, actually. And then in the Upper Jurassic, 150 million years ago, we already find species like this creature here, Haldanodon, with 180 degree of coiling, half of a turn. So this really looks like a nice transition. Yeah? It's a very easy story, but it's a bit more complicated. So all the groups between the monotremes and the therian mammals, they show a bit a puzzling pattern in terms of we do have some with a more or less straight or slightly bent cochlear canal, like this one here, or a more derived one with a straight one, so this is a bit puzzling. And then, coming closer to therians, we find actually a specific pattern of at least 270 degrees of coiling. And this nicely shows that there must be at least the evolution, or this coiling of the cochlea evolved at least twice in mammal evolution. So to sum up, we do see a kind of transition that takes up to date, actually 250 million years are represented here in this line. And this happens today in development within a few days. So there are studies showing in mice or also in platypus or echidnas that the coiling of the cochlea also is still visible in the early development of mammals. However, to point out, the coiling itself evolved at least 2 times. And why did this happen? So this is due to the fact that, of course, there is always a competition. So these Mesozoic mammals were mostly small, insectivorous, crepuscular, or nocturnal. They had to, of course, communicate but prevent predators. At the same time, singing insects might have been the proper prey for them. And there are reconstructions of fossil bush crickets, the noise they produced of the males to attract the females. And this actually does pretty much correspond to reconstructions of the frequency range of early mammals. So this is an idea we are just working on if there was a kind of acoustic co-evolution in the Mesozoic. The second example is about locomotory adaptations. So what we know from other studies, like this very nice comprehensive study of more of 200 terrestrial extant mammals, that if you measure the radius of curvature of the 3 semicircular canals and do some statistics in this kind, or in this case, regression analysis, so it's a mean radius of all 3 canals versus the body mass, then you get a nice pattern. And if you code these specific species with a kind of agility score or classification from fast to very slow, for instance in the sloths, then you can observe that more agile or faster moving species have larger semicircular canal radius than more slow species or slower species. So this is a very simple story. And what we did in a study several years ago, we took a closer look at xenothrans, especially on sloths, on both family of sloths, the 2-toed sloths and the 3-toed sloths. And we did measurements, or we did CT scans of the inner ears, the virtual reconstructions. And we also used some other species for outgroup comparison, like, for instance, here the European squirrel. And what you see here is 5 individuals of the respective species, and in the anteater, the armadillo, and the squirrel, it looks like copy and paste. But these are 5 different species. Instead, in the 3-toed sloth, Bradibus variegatus, it looks like 5 different species, actually. Yeah? And you can see here are examples for the extremes, that you have a different shape, size, diameter of the semicircular canals, or the planes of the canals differ in their angle, etc. We also did some statistics on this based on measurements, and as you can see here, the deviation is much higher in the 3-toed sloth than in all the other species. And also the PCA supported this in terms of that one species, which is coded here in red, of the Bradypus, covers a bigger space in this PCA than any of the other families we investigated. So what we can see here, sloths have a statistically significant variation in their semicircular canal morphometry, and this is due to the fact that they are doing nothing. They are just hanging around, so there is a release of evolutionary or functional constraint, which has already been described by Charles Darwin, that organs that are rendered useless may well be variable or even be lost. This cannot happen for the inner ear, but it works. It doesn't matter the shape, the size, it still works for the sloth. The last example is about the bony labyrinth variation in pantherined cats. And this is the work by my PhD student, Matthias Wirkner, who could unfortunately not make it today here. And he is interested in general in intracranial anatomy in pantherine cats, covering all extant species and also some fossil ones. We also published a paper with the first results on the comparative morphology of the bony labyrinth, and he's especially interested in inter- and interspecific variation, functional morphology, ontogeny, and also, in case we do have the metadata on the label of the specimens we investigate, wild versus captive or zoo animals. Are there any difference visible? And in fact, of course, here we have a high constraint on the inner ear, but we still found differences in the morphology between the different species, and we could clearly distinguish both living genera, Neophilis, the clouded leopard, versus the other pantherine species. In in the genus Panthera, we do find a structure called the secondary common cruise. This is simply the fusion of the posterior and lateral semicircular here in some part, and this is not present in Neophilis. It's also not present, based on literature, in other phalids, with some exceptions, and in most other studied carnivorans, which means that absence of the secondary common cruise might be a Grundplan character of pantherines and maybe carnivorans in general. Fun fact. The secondary common cruise actually is a primitive feature of mammals. It's present in all Mesozoic mammals that have been studied, and it has been lost several times independently in several lineages. So obviously Panthera shows a secondary common cruise. The function of this structure is completely unknown. It's just there or it's not there. We also did some simple statistics, more or less statistics here. For instance, we looked at the inner ear height of the bony labyrinth versus the body mass, or the anterior semicircular canal radius. And it's obvious that we find a negative allometry, which is expected for sense organs, nothing special. We found some specific patterns here in the clouded leopard we cannot explain, but we only have very few specimens in the study. So why both species differ so significantly, it's totally unclear. The negative allometry is also reflected in the ontogeny, the development, postnatal development of the bony labyrinth. And I should mention that in placental mammals, the inner ear, the bony labyrinth respectively, is ready to go at birth. So shape and size should not change anymore. And what we observe here, in fact, is that, the bony labyrinth is the same size in all these 4 stages, from the neonate to the fully grown lion. And we also found this pattern in the tiger and in the leopard. And only the surrounding bone, the petrosal, is growing. And there's also some other changes, like the small fossa here that houses part of the brain that is lost during ontogeny, which has already been described. And this size change, or this change of the thickness of the surrounding bone, is also reflected in this canal that has to reach the outer surface of the petrosal from the inner ear. That's becoming longer during these 4 stages we investigated. Then we took a look at the cochlear turns, and in general, our pantherines show 2.75 to 3.25 turns of the cochlea. Most of them 3 turns, so this is a very common pattern among many mammal species. But the few specimens in lions, tigers, and in the leopard, we could distinguish between wild and captive animals. We saw some differences. And in the lion and in the tiger, the captive one had normally, or had larger, or no, had more cochlear turns than the wild ones. And in the leopard, it was the other way around. And we said, oh, wow, we found some pattern, different pattern between captive and wild animals. Could be interesting for zoo people. But then we took a closer look, and obviously, neonate or juvenile specimens showed the higher degree or the higher number of the coiling. Is this an interesting pattern? So we are still working on that. This is in progress. But I can tell you, we are biased by a very small number of specimens for all these stages. And even if the label says the specimen was donated by a zoo, you don't know in older historical specimens if it was caught in the wild. Yeah? So there's no information about that. But what is known in the cat, for instance, or in the short-tailed possum, that there's a higher variation in the bony labyrinth in captive animals. And we did some other measurements and calculations on the so-called cochlear aspect ratio, which means that shows a difference in tightness of this coiling. You can have the same length, but a looser coiling leads to a lower number of cochlear turns and the tighter coiling to a higher one. And so we found some differences in this aspect ratio. And we also found a publication on the artwork on the ontogeny of the bony labyrinth that shows the same pattern, that the younger stages show a higher number of coiling than the fully grown specimens. And maybe there's something going on that the system, the surrounding bone that is still in the progress of ossification might somehow shape the change the shape of the cochlea or of the bony labyrinth in some respect. But this is still, as I said, work in progress. And by this, I want to thank you for your attention. And for those of you, unfortunately only in the audience, who are interested in a hands-on session on bony labyrinths, I have done some 3D prints of the bony labyrinths here with me. Thank you.
Can you hear me? Yes. Thank you, Irina, for this nice presentation. I guess we have time for one really short question. Yes.
It's a short question. So, in that plot where you were comparing mammals with birds and reptiles and their hearing capacity, I was curious because where do owls fit in that spectrum? Do you know?
Actually, this is not my work. I just used this figure and I did not check for the species, to be honest. But there's an overlap between mammals and birds, but it's a very small area. But where the owls actually fit in, I cannot tell you.
Okay, all right. Thank you.
I have to check the paper, actually. Sorry.
Yeah. I guess that's it. Thank you again for this nice presentation. One applause again. And we are continuing with our next speaker, Dr. Genevieve Housman from the Max Planck Institute for Evolutionary Anthropology. And she's a geneticist interested in primate functional genomics, complex trait evolution, and skeletal development and maintenance. In 2023, she started the Skeletal Genomics Research Group at the Max Planck Institute and for the Evolutionary Anthropology, where her team still continues to work with. The name of the presentation is Primate Cell Culture, Systems for Functional Genomics Research. Yeah, you can go on.
Thanks so much. Hi, everybody. I'm excited to be here today to talk to you a bit about primate cell culture systems and how we can work with them to examine functional genomics. So I have to confess I'm an anthropologist, and I find primates really awesome. So today's talk is going to be focused just on primates. I think primates are so cool because if we look across different species, there's so much phenotypic variation that exists between different species and also within species. And in addition to sort of characterizing that phenotypic variation that we see, what my group is really interested in trying to do is to not just characterize it, but to understand the mechanisms that contribute to that phenotypic variation. So when we think about complex traits, which are typically the ones that we're interested in for different species, they are in fact complex, which means that there are many factors that contribute to the development and the maintenance of these features. Genetics definitely plays a role, so your underlying genome sequence variation, but that's only one factor contributing to these traits. Another really important factor is how that genetic variation is actually expressed in different cells and tissue types. So how your DNA can be regulated is through chromatin accessibility, how open or closed the chromatin is, whether there are methyl groups bound to certain parts of your DNA that can impact whether certain genes are expressed or not expressed. Then we can think about genes themselves, and if they are expressed as RNA molecules or transcribed as RNA, what levels they're transcribed at, and then whether that RNA ever eventually becomes protein. And if it does become protein, are those proteins activated or are they degraded right away? Oh, sorry, those were supposed to show up. So to summarize, your genetics is really important for understanding traits, but it's also really important to measure aspects of gene regulation, because that will give additional insight into the mechanisms that are allowing certain traits to develop and evolve. And as I just mentioned in the previous slide, there are lots of different molecular phenotypes when we think about gene regulation, and so that adds a certain level of complexity. We have to look across many samples to measure all of these different phenotypes. And then one last complexity is that molecular phenotypes can vary across different contexts. And so to give an example of that, if you think about your own body, you have one genotype. The same DNA is in all of the cells that make up your body, but that DNA has to be expressed differently in different cells. So a brain cell has some parts of its DNA being expressed and other parts not, versus a heart cell. So the DNA has to be regulated differently to create all of the different cells in our body. DNA can also be expressed differently across different developmental stages, from early development to later aging. And then also it can change in response to different environmental exposures, our diet, activity levels, whether we're exposed to pathogens, et cetera. And so because of all of this complexity with molecular phenotypes and gene regulation, really the best way to study gene regulation and how it contributes to traits is to sample lots and lots of tissues from lots and lots of individuals and lots of species. And I don't really want to do all of that sampling, nor is ethically that an okay thing to do. But still, at the end of the day, we want to know mechanistically what's going on in all of these different contexts across different species. And so the big question is, how can we do that? And what I'd like to argue today is that cell culture systems are a really good method to move this type of research forward, that they can serve as appropriate approaches for recovering functional genomic information from different individuals and species. So for those of you that are not as familiar with cell culture, I'll just give you a brief overview about what it is. Essentially, it is the growth and maintenance of cells that have been isolated from animals in an artificial controlled environment. So basically, we're taking cells out from an organism and putting them into a petri dish. So to give you just maybe some pictures of some actual cells. This is what they look like growing in their dish. Sometimes they stick to the dish, sometimes they float in the dish. They are alive, they grow, they proliferate, and so we have to give them media that contains nutrients. We have to keep them in incubators so that they stay warm and that they can have certain gas exchange. And most importantly, we have to work in very sterile working conditions. So that's just an example of us working in a biosafety cabinet, because all of the conditions that we need to make sure that the mammalian cells can grow and be happy, those are also really great conditions for bacteria and fungi. And those are usually things that we don't want growing alongside our mammal cells. So to give a little bit more resolution on different types of cells that people like myself and others can work with, there are primary cell lines, and these are cells that are isolated directly from tissues and that can be grown in a petri dish. And typically, these people work with fibroblasts, which are derived from skin, peripheral blood mononuclear cells, also called PBMCs, which are derived from blood cells, and then mesenchymal stem cells, which usually come from bone marrow but can come from other tissues as well. And these are really great kind of initial sources for cell lines because you can get them straight from tissues, but they're finite. So they will proliferate for a certain amount of time, but at some point they'll sort of tucker out and not grow anymore. So they have a limited lifespan. So the next best cell line to work with is one that is immortalized, which means that it has some transformation that it's undergone so that it will continue to proliferate forever, indefinitely. So the main types of immortalized cell lines that people work with are either lymphoblastoid cell lines, which are immortalized immune cells, or cancer cell lines. And these are really great, again, because they can proliferate forever, but they have their own sets of limitations. So the best of the best cell lines to work with are those that are both immortalized and pluripotent. And pluripotent means that the cell has the potential to differentiate into lots of different cell types. And so these include things like embryonic stem cells, and then my favorite, which are induced pluripotent stem cells. And induced pluripotent stem cells have all the characteristics of an embryonic stem cell, except they're derived from adult cells. So they're reprogrammed to be like embryonic cells, but are not from embryonic tissue. And because immortalized and pluripotent cells are, I think, the best to work with, those are the ones that I typically work with. Oh, and sorry, I forgot to mention the iPS cells are awesome because they can differentiate. So this is just a small handful of different cell types that have been derived from different human and non-human primate iPS cells, bone cells, cartilage cells, brain cells, and cardiomyocytes. And I'm not sure how to make the thing at the bottom play, but cardiomyocytes are heart cells, and you know that they're heart cells because they actually start to beat in a petri dish, which is pretty cool. But yeah, so because immortalized and pluripotent cells, I think, are the best to work with. Those are the ones that my group typically works with. Sorry. Oh, oops, went a little bit too far. So yeah, before I tell you a brief overview of some of the areas that we're doing research in, I just want to reiterate one more time why I think cell culture systems are so useful. And it's really because they are in vitro systems, meaning that we're working with these cells in a lab. We can perform lots of different experiments with them in the lab, but leave animals to their own devices and not interfere with animals and invasively sample from them. When we derive these cell lines from animals, they retain their genetic information, so the cell lines are specific to individuals. And then depending on the cells that we're working with, we can theoretically differentiate them into lots of different cell types, and we can expose them to lots of environmental perturbations. And the list goes on and on. There are, of course, some reasons why you might not want to do cell culture, but overall, I think the pros outweigh the cons. I should note, too, that working with and establishing cell lines from primates is not something that is new. It's been happening for a long time, and I've reviewed some of the cell lines that have been established. Most of them are primary cell lines, so they have finite lifespans, and actually a lot of those lines are no longer available for people to work with. But there are a decent number of cell lines that have already been established for different primate species. But one of the big problems with these lines so far is that there aren't really unified panels of cells from many individuals and many species, and that have been derived using the same protocol. So they're sort of made here and there without a unified process, which makes it really hard to compare across all of these different systems. So what my group is really interested in doing is both, of course, working with cell lines that have already been established, but trying to also expand the number of cell lines that exist and generating them in a way that then allows them to be used as larger panels together. So I'll just briefly give an overview of the types of research that we're doing. I'll start with work that we're doing with previously established cell lines. One area that we're focusing on is using these previously established cell lines to generate difficult to access cell types. And my group is particularly interested in mesodermal cell types. So for this work, we've primarily been working with a panel of human and chimpanzee cell lines. It's about 7 humans and 7 chimpanzees. And this panel of cells was established at the University of Chicago, and we have obtained CITES permits to bring those cells to Leipzig. What we've done is we've taken these induced pluripotent stem cells, and we've been able to differentiate them towards mesodermal cells by first pushing them into this mesenchymal stem cell state. And then once they're in that state, then we can further differentiate them either into bone cells, which are shown here, and these are calcium-producing cells, and the calcium-containing matrix that they're producing is here stained red with alizarin red staining. We can also differentiate them into chondrogenic cells or cartilage cells, which are cells that also produce an extracellular matrix, but that contains collagen, and that's stained here with blue, a blue coloring. And we can also differentiate those into adipogenic cells as well, so those are fat cells. And they're, here we have them stained with oil red O, which stains the lipid molecules red. And right now we're actually working a little bit more to try to differentiate these a little bit further to specifically brown adipose cells. And in addition to differentiating these cells and then looking at regulatory pattern across all of these different cell types, we're also really interested in looking at regulatory patterns across relevant contexts specific to these different cell types. So we look at how regulation changes across differentiation of these cells, but we also look at regulatory patterns in these cells when they're exposed to different environmental contexts that are relevant for the cell type that we're working with. So for skeletal cells, we think about how mechanical stress on those cells impacts regulation and how other things like inflammatory cytokines impact. And for brown adipose, we're thinking about cold exposures. So to just give you an example of some of these studies, for stages of differentiation, what we do is we differentiate the cells, and to look at different stages, we just collect the cells at different time points and characterize the regulatory patterns. But one really exciting direction that we've started to go down is when we're making these organoid models, what we've discovered is as you're trying to differentiate them, not all the cells differentiate at the same rate. And so actually you can get heterogeneous populations of cells in your organoid. So you can have a clump of cells that has many stages of differentiation all at the same time. And what we do is we then use single cell sequencing methods to look at regulatory patterns in individual cells to kind of characterize that stages of differentiation all from one system together. I mentioned we also can do these perturbation studies with our different mesodermal cells. And there we can look at how regulatory patterns compare between control conditions and perturbed conditions. And when we have large enough sample sizes, we can go one step further and not just look at gene regulation responses, but also how does underlying genetic variation either predispose individuals to having certain regulatory responses or protect them from having certain regulatory responses. So if you're familiar, those are quantitative trait loci. So in addition, sorry, let me get a little water. In addition to perturbation studies with the skeletal cells, we're also really interested in exploring perturbation studies more broadly. And for this, I'll just very quickly mention we're doing genotype by environment interaction studies in larger panels of cells, specifically lymphoblastoid cell lines. Here, to start, we've just worked with a few individuals and cell lines, but we have a panel of over 100 cell lines that we're working with, and we're exposing them to many different environmental perturbations, immune, hormone, stress, nutrient, pharmaceutical drugs, and then looking at regulatory responses. And we do this using traditional cell cultures where different individuals are cultured separately, but also we are conducting what we're calling phylogeny in a dish, where we co-culture individuals and species cells together, and this allows us to scale things up much more dramatically. And so we're conducting these experiments and looking at regulatory responses, which is really interesting because we can both see how gene expression patterns vary across treatments, but how that varies across different species and what responses are conserved and what are divergent. So I mentioned work that we're doing with established cell lines, but I also briefly want to mention that we are generating new cell lines, both lymphoblastoid cell lines and induced pluripotent stem cell lines. And we're doing this in partnership with the Leipzig Zoo, and specifically the Wolfgang Kohler Primate Research Center, which houses all of the great ape species. And we have a long collaboration with them that when animals undergo necessary medical procedures, anytime there's leftover biological samples, typically blood, they are willing to share that with us. And from that, we can establish lymphoblastoid cell lines and then reprogram those into iPS lines later on. So here's just a couple examples. And then I know I'm running out of time, but I just briefly want to mention that we have started to do a completely non-invasive sampling method where we're collecting urine from the different primates. So primates every day wake up and they pee on the floor, and then we go in and we pipette their urine off the floor. And every once in a while, there are epithelial cells, essentially skin cells, that get sloughed off in that urine. And when we are able to get a sample that has that happens in it, we can actually get cells from their urine and make these cell lines. So this is the first line that we established. We have another one that we just established recently. And I just want to mention that this line was established from an individual named Keo at the Leipzig Zoo. And actually last week, he unfortunately died due to some health complications. But because we were able to establish a cell line from him using completely non-invasive methods, we can still get a lot of information about regulatory landscapes across all of his cells. And that also meant that usually for functional genomics research, if you want to look at that in the tissues from primates, you need to sample those tissues really, really quickly after death. And in this case, because that wasn't a necessary thing to do, the zookeepers were able to allow Keo and his body to stay with his relatives for some time so that there could be a mourning process. And at the same time, we're still able to work with cells from Keo and learn a lot about his biology. So we're doing a lot of cell culture, functional genomics research. All of this is to be able to try to characterize regulatory landscapes across as many contexts as possible with as minimal and non-invasive methods as possible, all with the goal of trying to understand the mechanisms underlying traits. And I'm just one group working on this. There are many other people working on it, but really it's a community effort. So if any of this type of research is of interest to you, please come chat with me. With that, I'll thank everybody in my group.
Thank you, Genevieve, for this nice presentation. Hello. Are we already connected to Denmark? Er du her? Jens?
Yes, I'm here.
Okay. Just let me say something and then we are continuing with you. Yes, any questions for Genevieve in that room? No. Also for the online participants, we are collecting your questions because we had some problems with the live stream. And we are telling the speakers them afterwards and then they will contact you probably. Yeah? Thanks. Yeah, our next speaker is Dr. Jens Svenning from Denmark, from the Department of Biology at Aarhus University, Denmark, yeah, like I said. Jens Christian Svenning is a macroecologist and biographer with a strong interest in plants, animals and people across the world. He's a professor, like I said, at the University of Aarhus, where he also serves as the director of DNRF Center of Ecological Dynamics in the novel Biosphere Econovo, established in 2023. The name of his presentation is Trophic Rewilding as a Restoration Approach Under Emerging Novel Biosphere. Thank you. Have fun.
Thank you for the introduction. I hope you can see my slides as they should be. It's really a pleasure being able to present to you here at the conference, and of course I'll take quite a different perspective on evolutionary diversity relative to the animals that you're all working on. So the topic for me is trophic rewilding as a restoration approach under emerging novel biosphere conditions, and I'll unpack for you what this all means in the following. So first of all, the big essential question that global society is facing is how do we maintain a livable biosphere in the future? And this is a super big challenge. It's not easy to deal with, certainly not in current political circumstances. So the key issues that we are dealing with is limiting climate change as much as we can, and that means limiting greenhouse gas emissions. If we want to overcome the biodiversity crisis that I'm sure you all have on your mind, it's very clear that the basic solution is to upscale and upgrade space for nature. The upscaling part is what you can see illustrated on the right. One of the fundamental relations in ecology affecting global biodiversity is the species-area relation. So the greater the area, the more species, and vice versa. And that simply means that it's impossible to maintain Earth biodiversity in the long term just on small areas. Of course, you can do that as an emergency short-term solution, but in the long term, that's not going to work. At the same time, if we upscale space for nature, that will have massive co-benefits for climate, because natural ecosystems will sequester much more carbon than managed ecosystems do, and also there will be big you can say advantages for quality of life. For example, if you look at the positive effects of green space on mental health, which is super well documented. At the same time, as a third part, you also have to maintain and improve human livelihoods and well-being, because otherwise you don't get support for doing the other things. And of course, also from a more basic ethical perspective, that's something that's important to do. And this interplays with maintaining free democracy. I don't think we'll get any reasonable solutions to the to global sustainability without that. So, and you risk that if people get too unsatisfied, they will vote for you can say non-democratic forces. You all know that. So another point I want to make is that when we think about biodiversity conservation, stasis is not an option. And what you see here on this diagram is in the top panel, you see atmospheric CO2 concentrations, and in the bottom panel, you see global surface temperatures. And then if you look to the right, you can see where the line starts to diverge future scenarios, and how they develop depends on our emissions. So far, we are somewhere between red and yellow, more close to red, to high-end scenarios. If we look back in time, already now we have to look back about 4 million years to find as high CO2 levels as we have. That's super long. That's super far back in time, very different world. If we look into the future, both for atmospheric CO2 concentrations and associated surface temperatures, we may have easily to look back 15, 30 million years to find something similar. And I can tell you, Germany, for example, 15 million years ago had a climate like southern Florida. So that means extreme changes. Everything in terms of natural ecosystems will change if that happens. I also want to highlight that this is not just scenarios. So this graph here is from the latest IPCC report. And if you look at this violet dot out to the right, that's the global surface temperatures for the last decade. And then you can compare that to temperature over the last 10,000 years, which you have here, which is or 12,000 years actually, which is roughly the whole history of civilization. We had innovation of agriculture here at the start. And you can see current situation is warmer than any time in the history of civilization. So we're already in unprecedented conditions. When you are in Germany or in Denmark or many other places, it can be hard to see how do we make space for nature. And this paper that came out in the journal Nature Food a couple of years ago highlights one very feasible way where we can do it. It's a paper, an analysis where they show that if the 54 high-income nations, the richest countries on the planet, they would shift to basically a flexitarian diet, very plant-based but not vegetarian. One could reduce the agricultural area by more than 400 million hectares, the whole size of the European Union. It wouldn't be all located here, but a huge area that wouldn't be needed anymore for agriculture, while still maintaining healthy diets for people. So that's just that. And they in this paper looked at the massive climate benefits to that. But if this was done in a smart way, this would also have massive advantages for biodiversity, of course. So I talked about upscaling and upgrading, and the rest of the talk I want to focus on upgrading. And upgrading is basically equivalent to what I would term rewilding, and you can see the definition here in green. That's restoration to promote self-regulating complex ecosystems through, on the one side, restoring natural, non-human ecological factors and processes, so the functionality, you can say, out in natural ecosystems, and on the other side, reducing human pressure on these systems. So you need both of these things, and there are many things more to say about these different dimensions to consider. One of them is trophic complexity, which is food web complexity, the complexity promoted by the animals, and that's what I'll come back to. One of the advantages to this approach is that it's focused around natural processes. It means that it's autonomous. It doesn't rely on people doing all kinds of maintenance for now ever on, which will, of course, not happen in the long term, will not be effective in the long term. We already have a scarcity of people in many places from the global demographic transition. Also, lots of people don't want to work out, you can say, in the countryside in many places. So it's not really feasible to go that way. But of course, natural processes have generated the diversity that we have over millions of years. So restoring it is a long-term scalable solution. The part that's around restoring food web complexity is what we call trophic rewilding, and that can be defined, as we did in the first paper about this in 2016, as species introductions to restore top-down trophic, so food web interactions, and associated multi-level food web interactions, trophic cascades, to promote self-regulating biodiverse ecosystems. This doesn't inherently only consider large animals, but it's very much focused around large animals because of their selective loss worldwide, as you all know, and because of their ecological importance when they are allowed to be there, you can say, in the ecosystems. And you can sum up the kind of effects that they have and how this works in relation to what trophic rewilding is trying to achieve in this diagram. So you have different guilds of large-bodied fauna, megacarnivores like lions, mesocarnivores, meso-herbivores like roe deer and white-tailed deer and so on, big herbivores like bison, giant deer, moose, dromedary, so forth, and mega-herbivores, which are the proboscideans, elephants, the rhinos, and so forth. They generate a range of processes. And the bigger the arrow here, the more important, because the bigger species actually at the ecosystem level generate the bigger processes when they're allowed to have their natural abundances. And these processes are browsing, grazing, nutrient and seed dispersal, carbon pool diversification. That's the generation of specialized organic microhabitats such as dung, living bodies, dead bodies, which many other species depend on, and non-trophic disturbances, so you can also call it erosion. And those things together, on the one side, generate heterogeneity, that means variation in vegetation and soils, and other aspects of the abiotic environment, and on the other side, and have this enhanced dispersal. And heterogeneity and dispersal are two things that very consistently enhance biodiversity, not just at local scales, but also at bigger scales. So restoring the functionality of the fauna this way increases the diversity capacity of natural and semi-natural areas. That's the whole idea. This is a big field study that meta-analysis that we did of field studies around the world last year, where you can see some of the empirical evidence for some of these effects. It's nearly 300 studies from around the world. This is a summary here, where the green arrows, they show positive effects, and violet arrows show negative effects, and thicker arrows, bigger effects. But you can see how large herbivores in these field experiments, where they are excluded from natural big ecosystems, have effects on everything from plants to soil nutrients, to soil structure, to litter cover, to bird abundance and diversity. And because the individual of these nearly 300 studies, of course, have replication, we also look at how they affect local spatial heterogeneity. And the green arrows here show that they promote heterogeneity in plant cover, plant biomass, small mammals, birds, invertebrates, but also consistently bare ground, which is actually a good thing. I'm happy to expand on that at some point. I want to highlight from a practical level, if we look in natural areas in Europe, and we did that using satellite data here, we see a very widespread densification of the vegetation. All the green areas here, that's where the satellites show that natural areas are densifying from woody expansion, but also expansion of coarse grasses and big forbs. And it's clear that this is very strongly linked to the loss of large fauna or absence of large fauna at functional diversities, functional densities in our natural areas. So it's a super widespread problem because a large part of our biodiversity in Europe, for example, depends on open and semi-open habitats. And if you look to invertebrates, also like on bare ground and these kind of more microscale openness structures. We also have many field studies from European trophic rewilding sites that show that if we restore the fauna, they very effectively restore this functionality and counteract this densification. This is from a famous rewilding site in the Netherlands, a site that without the herbivores would be completely covered by dense woody vegetation and dense reed vegetation. And as you can see, that's not the case thanks to the herbivores. So that's just one example. I also just want to illustrate these kind of biotic microhabitats that the herbivores generate, and that if we restore them, that they also allow to continue or recover the living bodies. Like you can see illustrated here is a photo of actually my own leg, where I discovered during fieldwork in a South African national park this big tick, which is a tick that is obligately dependent on rhinos, for example. Horned dung beetles below, super rare nowadays in Northern Europe because they depend on big dung in nature. And here a bone skipper fly, which was thought extinct in the 1800s in Europe, but was rediscovered in a small part of Spain, where they thankfully don't clean up the cattle carcasses as much as we do everywhere else. And this species is obligately dependent on old ungulate carcasses, just another example. And then I also want to highlight a more subtle thing. Large animals are much more mobile than small animals, and for that reason, larger species are much more effective in dispersing plants. And you can see the scaling here from a big study where we cut together bird and mammal plant frugivore interactions, mammal animal mobility and seed passage times, and linked all that. And you can then see here very strong scaling between the dispersal distance for plants by animals to body mass. So the bigger species of mammals, they actually disperse on the daily basis plant propagules much further than birds. And they actually also are the ones that disperse the biggest variety of plant species. So they're super important for that. The other figure here just shows that very generally, there's enhanced germination for plant seeds if they pass through an animal. So those two things together means that large-bodied species are super important for plant migration, which is very important for maintaining local plant populations, but also for plants tracking climate change. And this map here, just look at all the red color. They show all the places where historical losses of the fauna have really reduced this kind of plant migration capacity. So that's a super widespread phenomenon. And the blue areas are areas where it's pretty well maintained, but it's at high risk because the species doing the dispersal are endangered. Yeah, this is apart from moving plants, large animals are also super important for moving nutrients around between the sea and the land, and between aquatic ecosystems and land ecosystems, and within land ecosystems, because of this mobility and because of the historical and ongoing downsizing of fauna. These percentages that you can see here on the diagram, they illustrate how much this capacity for moving nutrients around have really been reduced. So also in this way, there's a strong functionality coupled to the large animals. Yeah, and the final thing I wanted to highlight is that related to the large animals, I want to highlight what I see as a big blind spot in conservation in Europe, namely that the ox and the horse are native large-bodied herbivores in Europe. They have a history of more than 500,000 years as wild species in Europe. They are not extinct as species, but we don't have any kind of international program for effectively restoring these species as wild-living, evolutionary evolving species in Europe. And this is really a blind spot that we should take on. And for the horse side of things, we just published this paper taking this perspective if you're interested. For the bison, for the European bison, it's realized, of course, that it's a native species in Europe, but it's very much misunderstood as a species of closed forest, while actually it thrives much more in mosaic landscapes and also help to generate and maintain them. So it's important to renew our picture of this species. Yeah, I'm just going to skip this, and just to sum up, there's a strong need for upscaling and upgrading space for nature. There's a key role for trophic, so food web rewilding, especially large herbivores, also large carnivores, I didn't have time to say that, to enhance biodiversity value and climate resilience of wild and semi-wild ecosystems by promoting vegetation and soil heterogeneity, by generating bioarteric microhabitats, and by enhancing dispersal processes. There's a lot of empirical evidence for that this actually works in the real world from trophic rewilding projects. And it's based on general mechanisms that we also can expect to work in a novel future where the climate will be deviant from what we're used to, where there's more and more biotic globalization, where species come in from different parts of the planet. These are general mechanisms, they will still work. And with that, I just want to thank you for your attention and thank all my collaborators and funders.
Yeah. Thank you very much for this beautiful presentation. Do we have any questions here from the audience? Yes.
Hi, hello Jens. So my question is, so I work in zoo research, and often zoos are criticized because we only keep megafauna, elephants, tigers, bears, etc. And most species saved from extinction is like the Szewalski horse, the Arabian oryx, etc., etc. And kind of the small little creatures are a bit forgotten because it's not what brings people into the zoo. But listening to your presentation, do you think this is an unfair assessment or critic towards zoos?
It's a somewhat biased assessment. I should also say that I'm actually very much a plant-oriented person and scientist, so I appreciate all the species that are not big species. But the fact is that over recent prehistory, over historical times, and what we see also now, there's been a huge selective loss of the bigger species from ecosystems worldwide. There's been huge extinctions, but huge range declines, huge abundance declines, and at the same time, they are very important for the functionality of the systems. So for that reason, zoos play a big role in avoiding that these species are lost totally, and also, of course, to make the public aware that these species are species we should be thinking about restoring. So I think it's not wrong that zoos have attention to these species, but of course that doesn't mean that it's not also worth having attention to other species, but I don't think it's misguided. And for example, if you look at conservation status of extant mammals, the bigger species have a much higher average risk of extinction than the smaller ones. It's not to say there aren't small ones that are in trouble, but there is really a size relation there. So I don't think it's overall wrong to have this kind of bias, you can say.
Thank you very much.
Okay, Markus Klaus just has one short question. Yes.
Thank you for this really nice talk, Jens. My question is, I see this parallel between people not wanting to kill animals in zoos, and then you have these problems with the rewilding projects, especially the one that you showed. I can't pronounce the name. What is your message or your strategy, how these projects should be regulated in terms of letting animals die or harvesting them by hunting? What is the strategy you propose?
Well, I think it's it's certainly a tricky issue. I mean, first of all, I mean, most people don't like death. I don't like death particularly, right? But at the same time, it's a super important, mortality is a super important natural process. And I mean, you can think about natural selection wouldn't work without mortality. Horses wouldn't look like horses if there hadn't been very strong mortality processes causing that morphology to be successful and so on, right? So mortality is super important in nature. And I think there's a communication aspect around that zoos can play a role in. In practical nature conservation, I think there's a scale issue. It makes sense if you have, you know, a couple of horses in a few hectares, you have a huge responsibility for those. If they have access to a huge landscape, then I think you should explain to people that it's okay that they die from whatever reason. It's natural, and part of that also, it's not only natural to die from predation. You can also, and it's also not necessarily nice. You can very often also die from food stress and climate stress. I mean, like the mega herbivores, they're not top-down controlled. They mostly die of starvation when they're too old or something like that, right? That they can't, their teeth are destroyed or something like that. So there's something to explain to people. I mean, it also happens for smaller species. I mean, like our overwintering birds, for example, there's huge mortality in winter for those, but people don't think about that the same way. But I think one has to have a continuous approach. Of course, if the animals are super constrained, it's hugely your responsibility. If they're basically not more constrained than what is natural, then I think we shouldn't interfere in the mortality processes. And then there's a gradient in between there where I think it would be nice if our legislation could be adapted to deal with that complexity in some way.
Good. Thank you very much, Jens. I guess that's it with the questions. Also, thanks for participating at our conference. Greetings to Denmark.
Have a good conference.
That's it for session 1. We have now a 5-minute refreshment break until 11:30. So for all the online participants, you can check out and check in session 2 then. But before that, before you going to the toilet and so on, we forgot to hand over some presents for our invited speakers. Markus Klaus, of course, Zorana, and Genevieve and Irina, of course. These are some nice goods from this region, like honey, tea, and so on. So have fun with it. Yeah, and see you soon in 5 minutes. Now we have a different session, which is more focused on the topic conservation. And our first speaker on this session is Professor Dr. Werner Kunz of the Heinrich-Heine-Universität in Düsseldorf. Professor Kunz is, or was still, a zoologist and molecular biologist with philosophical interests. And in order to promote endangered species, he argues for a strict separation of species protection from nature conservation, and calls instead for the creation of special biotopes that should be set up by the farmers and foresters to meet the needs of specially selected endangered target species. And the name of this presentation is Reintroduction of Species that have become extinct due to habitat destruction in Central Europe. So, Werner, stage is yours.
Das ist bei...
Das ist der Pointer für nächste Slide und hier ist ein Pointer.
Und das ist rückwärts. Das ist rückwärts. Ja. Yeah, thank you for the invitation. I think my presentation will be a bit unusual because I'm speaking mainly on insects, and in zoos you usually do not find insects. The title is Reintroduction of Species that have become extinct in Central Europe due to habitat destruction. I give you examples where you can save endangered species by rewilding them after having increased their number by culture. Some examples of species that have been saved from extinction through breeding, habitat management, and reintroduction into the wild. Everybody knows the hamster, which is very rare in almost all the countries of Central Europe, and there are some successful rewilding examples. This is an animal, the bald ibis, that was extincted already 400 years ago, and it's now rewilded. But also there are some butterfly and other insect examples, like here, the golden fritillary. So my presentation concerns on saving endangered species in Central Europe. Why? This is the main question. Why have so many species become rare or even threatened with extinction in Central Europe today? What can be done? The number of human beings on Earth exceeded the 8 billion mark 2 years ago, 3 years ago. And this obviously is the main reason why we lose so many wild species. And I show you an example which I have found in Google, which explains almost everything. This is how the world looks like today. But not only human beings, but also the animals that are reared by humans. We have now around 1.5 billion cattle on our planet, and we have around 25 billion chickens on our planet. This undoubtedly, as I mentioned already, is the main reason why so many species are becoming extinct. But this cause is not mentioned in the first place. They are coming with climate change and with poisoning, but this is the main reason. The decline in biodiversity in Germany goes on and on. A visible turnaround is not recognizable, despite so many nature conservation areas. We have so many species. Why have so many species become rare or even threatened with extinction in Central Europe today? Today in Germany, we only have 20% as much biomass as in 1850. Not only the number of species, but the whole number of individuals, the bird mass. This is a loss of 80% in the last 2 centuries. And with the insects, this is even worse. We have a loss of 80% insect biomass in the last 30 years alone. So if we used to have a certain area, So if we used to have in a certain area 100 insects in earlier years, then today in the same area there are only 20. Most of the pictures are my own photographs. This is a very, this says everything. A European Environmental Agency, the Grassland Butterfly Index in Europe, and you see, particularly in the last 6 years, that it goes down. Drastic declines in butterflies, especially in the last 10 years. What has happened, particularly during the last 60 years after the Second World War? There are, of course, several causes. I am focusing here in my short talk on one of these causes. We now have a landscape in Central Europe that we never had before. What did the landscape in Central Europe look like in the past? And what it does look like today? Let's go 2 million years in the European past. Large areas of Central Europe were for more than 2 million years during the glacial periods conserved by the mammoth steppe. Many people believe that the mammoth steppe of the glacial periods was not habitable for many species because of the permafrost soil. But exactly the opposite was the case. The mammoth steppe was a very species-rich habitat, also e.g. for the cold-blooded butterfly species. This is a reconstruction of the mammoth steppe. You see big animals, but you also see particular insects that are threatened by extinction today. This is what the forest looked like during the interglacials, between the glacial periods. It was not a dense forest. It was a very open forest, and the steppes of the glaciers were kept clear by the large animals and mega herbivores in the forest areas. There was almost nowhere this kind of dark forest with close canopy. In earlier millennia, the forests were always much higher, much lighter, and with several sunny spots on the ground. But at the end of the last glacial period, the mammoths were wiped out and other big animals also. So we lost most of the mega herbivores during the which were living here in the Pleistocene. In the last interglacial, there were a lot of mega herbivores. Here are the examples. And today, in the Holocene, only very few of these species are still alive. The mega herbivores were gone in the Holocene, also in the now present area, but humans, now sedentary, took over the role of the large mammals and defied reforestation. Man, instead of the large mammals, now kept the landscape open and prevented it from becoming overgrown with dark forest. This is by several publications proven by the pollen analysis. Man made large fire and kept open the forests, For thousands of years, until 100 years ago, the Central European landscape was always an open land. Not so densely overgrown with grass as today, not so overgrown with shrubs as today. The forests were open forests with sunlight at the ground. This is a historic photos, a historic utilization. The forests were cleared because they need the soil for the cow shed. Here another historical picture, so-called Plaggenwirtschaft, removal of the human layer as bedding for the cattle sheds. Why did the landscape in Germany look like? What did the landscape in Germany look like in the last centuries? This is a picture from 1842. It is the Ahrtal near Arenal, and you look, it is completely different than today. All was open. And you can imagine several butterfly species which were living there and can't live there anymore. A landscape in Thüringen. Nothing of this landscape is present today. These are the landscapes to which most animals in Central Europe are adapted, not to the dense forests. One thing is certain today, we lost our open landscapes. One of the main reasons of today's species decline in Central Europe is eutrophication, nitrogen fertilization from the atmosphere, and lack of, this is the second point, is very important, lack of utilization. All areas outside the agricultural lands are not used anymore. That's why today the landscape is overgrown. We have too much afforestation. 3/5 of the 436 European butterfly species use dry grassland as their most important habitat. The meadow in earlier times, a meadow of today, a wayside in earlier times, a wayside today, a forest in earlier times, a forest today. Several butterflies need open grounds, as here with the swallowtail. It lays the eggs in flying, and it can't lay eggs in the dense grass. Many butterfly species are close to extinction in Germany because they no longer have open grounds, no sparse vegetation. The blue butterfly, Poliomatus damon, is just dying out during these years in Germany, the last places we will lose this butterfly species. Also the hairstrake, Satyrium illicis, it was 50 years ago, there were hundreds in the country Saarland. Today there are only 2 places. The fritillary Euphydrias aurinia is all close to extinction. And today we can no longer save many species by letting nature run its nature. This is a bestseller in Germany, but this is the wrong solution for saving endangered animals. Man's wishful dream of an unspoiled nature does not save many species that are close to extinction. Today, mankind has the moral obligation, but also the technical means to restore the lost habitats. That is what conservation must, now the examples must look like in the future. This is a EU LIFE project, and they altered the landscape by restoration historical landscapes, and they say we restore the original landforms. In this way, heathlands and dunes are recreated in their original location. This is another example of the EU project. We create vegetation-free areas through controlled burning, topsoil removal. We recreate vegetation-free areas for flowering plants that can grow there. And their example is the fritillary, the golden fritillary, Aurinia. It has been saved, gerettet, by these landscape restoration. This formerly common species was completely extinct in Schleswig-Holstein. It could be successfully reintroduced through, first, breeding under artificial conditions, second, rigorous technical habitat management, and third, releasing the bred animals into the wild, like this, breeding the caterpillars and butterflies. Step 2, the forest had to be removed, which of course was only possible under protest of the population. Step 3, then the humus had to be removed. This also was only possible due to protests from environmentalists. At the end, the habitat looked like this. And step 4, the fritillary could be successfully reintroduced. The next example is the Apollo butterfly, which has a certain subspecies in the Moselle area, and it's now dying out. During these years, it was common 10 years ago, but now you see almost nothing. This butterfly is dying out. It's now to be saved through extensive conservation breeding. We have no other choice.
Yeah, thank you, Dr. Kunz, for this beautiful presentation concerning conservation. Do we have any questions here in the room for Dr. Kunz? Nope, no questions. Good. Then I would say thank you for your participation at our conference. We really appreciate it. That's why you get, of course, another present from our Lüneburger Heide. So, we're continuing with our next speaker. I guess everyone knows him. Jon Taylor, he's already there. Okay. Jon is a CEO of Conservation Optimism, and he has over 30 years of conservation experience, including years living and working in Africa and Asia, and roles with organizations such WWF, the Zoological Society of London, and Save the Rhino International. And to describe Jon Taylor, he said the sentence, he spends much of his time living in a van and wishing he understood humans better. Well, I guess you can continue on that.
Thanks. I wondered what I wrote. Yeah, my presentation will not be beautiful. It will be rushed. I've got a lot to cover. I've got 20 minutes. So the best I can hope for really is a table of contents. And I'm hoping that some of this is interesting and we can pick up the conversations later. And I have 20 minutes, so I'm going to hit start on the stopwatch. Thank you very much. I'm Jon. I'm an old zookeeper. I'm an old wildlife zoo conservation program manager. Emphasis on the old. I framed this in this way for a reason. The abstract says this is about positive communications, and to a certain extent it will be. But what I'm hoping to do, we've had some fantastic presentations this morning. Fabrizio asked my favourite question, which is wherefore? Wofür, I think. I told you I knew a bit of German. Why are we doing this? And he asked those great questions about why this relationship with other species and the rest of the world. Marcus's brilliant presentation this morning was asking us in a more challenging way, as people who run zoos, what on earth are we doing? I kind of want to ask some of those questions about conservation. So the reframing is important. A lot of the first bit has been said already, so I can flick through, but I'm concentrating on H. sapiens vulgaris. You understand this is the common human, or as we know it colloquially in the UK, muggle. This is muggle conservation. It is not about any other species, for a reason. Muggles have been around for a long time. They evolved through various, is this going to work, or is this going to be, yeah, it's going to work occasionally. They've been bumming around for millennia, just throwing rocks and sticks at each other and doing gentle farming and stuff. Eventually, various regional civilizations have bloomed, but they tend to decay away through internal decay or external violence. But in the end, global development picked up to a point where in some northern countries there was an agricultural revolution that freed up labor for an industrial revolution, and the world was in a place where that was just communicable enough to go global. And the population that you can see along the bottom, having bummed along for years, took up until 1805 to reach its first billion. It then took another 100 years, only 100 years, to reach its second billion in 1925, and the rest, as Werner has just pointed out to us, accelerated at a very quick rate. There are now 8 billion people on the planet. They are not all heavily industrialized, but a lot of them are. That has consequences. CO2 levels have bounced around between 150 and 280 parts per million for hundreds of thousands of years. That's just the Milankovitch cycle. We can talk about that later if you like. But they're now well up above 400 over the last 150 years. There are, as we heard last night, a whole load of plastics in the ocean. We are polluting the place. This is a thermal pollutant. There are lots of others. Those humans consume like nothing else. So the Living Planet Index, even since 1970, when some of us were actually alive, has dropped, is it 73%? Can't remember what the latest report is. I think it's 73%. So that's abundance of a whole range of species measured by WWF and ZSL. So that's plummeting, and we've already seen, 2 people have pointed out this morning already, and I'm going to be the third one, that wild mammals now make up 4% of global biomass, and the rest of it is humans and human domestic animals. And H. sapiens vulgaris appears to be overexploiting its environment. If you want to find out what happens to animals that overexploit their environment, go and look at northern lynxes and snowshoe hares. Given this audience, how many people know about northern lynxes and snowshoe hares? I hoped as much. Good. One's hands didn't go up. Which universities are you at? See me afterwards. We can go over exploitation of natural solutions. How much? Oh, God. Too slow. IUCN lists H. sapiens vulgaris as least concern. I challenge that classification. I would change it to vulnerable. They are overexploiting their natural resources. We know what happens when that happens. At very least, it tends to lead to conflict, pressure, and possibly the election of orange people to run very large and powerful countries, which isn't really good. Conservation is about H. sapiens vulgaris. It is not, and I'm not picking on anybody here, we love the butterflies, we love the primates, we love anything that has interesting ears, we love many, many things, but we love them. This is through a human lens. There are many human lenses, but we have to understand that we see the world through a human lens. There are probably many, many other ways of viewing the world through other lenses, but we cannot look through them. This is humans and our relationship with the natural world. And H. sapiens vulgaris is important. The main drivers of the conservation crisis, and indeed the climate crisis, are H. sapiens vulgaris. We can go out and study why bears wipe their bottoms on trees as much as we like, but what bears do with their toilet habits is not the big problem here. Big problem is Homo sapiens behavior. So if we want to do something fundamental about it, we probably need to be engaging with H. sapiens, and what we probably need is large-scale behavior change. The best way the only way I know of getting humans to change their behavior in a democracy, where you can't just tell them what to do, is through communicating. So we've got to talk a little bit about communications. And for a lot of you, your communications department is separate in your zoo from your conservation department, probably separate from your research department, probably, unless you're here, apparently, where they're all in the same department. Deep respect to you guys. We communicate, we do conservation. Let's collect some conservation euros and then send them off to some other country, and we can buy some boots for rangers or put traps out or whatever it is we want to do. And then we go and communicate about it because we want to raise awareness of what a wonderful zoo we are, or perhaps we want to do some more fundraising, so we go and talk about it. I argue that we are not communicating about conservation anymore. And those of you who have different communication and conservation departments need to think about this. This is communication for conservation. Communication is now our biggest conservation tool. Yes, we can go out and throw some quadrats around and try and clear up the biological mess behind H. sapiens. But the main job is actually engaging with H. sapiens in the first place. In America, I think that the expression is, they call it the third rail. There's a brilliant line from the West Wing. They don't want to engage with social security, and they say, oh, it's the third rail. They mean you've got the two rails in the train track, and the third one is the electric line. Don't touch the third rail. And one of them turns around and says, yeah, because that's where the power is. Engaging with H. sapiens is the third rail here. This is where the power is. No disrespect to last night, but we can have as many nice websites of conservation heroes as we like. Engaging with large-scale human behavior change is going to be how we really change things. I usually stick know your audience in because I'm talking about communications. And the other slide I normally stick in is know thyself. What I normally mean by that is you can't communicate people until you know what you've got to say. And that's an important point. But in this case, our audience is ourself. So know thyself has another meaning. We need to understand Homo sapiens vulgaris if we are going to engage with Homo sapiens vulgaris and get them to change what we're doing, what they're doing. Eek. 1 minute per slide. We could spend a whole presentation on any one of these models. This is an old one. I put this in because you probably you know Maslow's hierarchy of needs. This is Abraham Maslow, 1940s, 1950s. It's a very old model, but it's solid. It works pretty well. Humans will engage with the good stuff when the necessary stuff is in place. If the pressure's on, and they feel that they're under threat, the good stuff tends to go out the window, and we get back to the election of orange people again. This is not good. Well, unless you're a big Trump fan, in which case I apologise for my aspersions. I am not. It's a reasonable model, and we come back to it. But there are 21st century models of understanding humans. This is Shalom Schwartz's model of universal human values. This is social science. I'm sorry, but this is social science. Some people think that that's valid too. These are all the values we have. They think this is not what kind of person am I, it's just where am I operating at the moment. These values interact. There are different relationships between them. We can talk about that for hours, and I'd love to talk people through it, but I haven't got time. But it's important to understand this stuff if you're going to take people on a journey, if you want to move them from place X to place Y. The top stuff, for example, is all about self-transcendence and the possibility of self-realization. That's where we like to be when we're feeling good about ourselves. Yeah, we can make the world a better place. Love and peace, dudes. Put the pressure on, down. Put the pressure on, and we come bouncing back down here pretty damn quick. Self-enhancement, self-security, conservatism, not conservation, conservatism. Vroom, little red hats. This is I mean, we could just spend the whole day talking about Kahneman's work. Have you read Thinking Fast and Slow? Anyone? Good, some people have. 95% of our thinking is probably system one. It is unconscious. It is effortless. His whole work was about understanding the assumptions and heuristics that the brain employs to achieve that. It's brilliant. Humans have a brilliant automatic mode. Our whole scientific method is designed to circumvent that and all the biases that are involved in that. 5% of what we do is effortful, conscious, intentional, problem-solving thinking. And system 2 is lazy. It wants to make stuff automatic and put it into system 1 as quickly as possible. Anybody who thinks that the conscious mind is the sort of snow-capped peak on the top of the mountain is kidding themselves. The conscious mind is a small observation platform out to one side. Most of the mountain is unconscious. This is the animal we are dealing with. We think, therefore we are. No, we don't. H. sapiens vulgaris is a feeling creature. We feel, therefore we are. If you're studying advanced science, eek, or panicking at the clock, it's not at the end of the day because you think it's the right thing to do. You maybe think it's the right thing to do, but you're doing that because doing the right thing makes you feel good. We do the things that make us feel good. We try to avoid the things that make us feel bad. Homo sapiens is a feeling creature. If we're going to communicate with these things, I'll skip past him. We try and reach out with these statements as scientists. This is trying to get people to change. Rapid and far-reaching transitions. IPBES. Transformative change and a complete re-evaluation of our profound shift of our relationship with the natural world. We are trying, what are we trying to do by putting these kind of things up? And we put 20, 30 deadlines on them. We need to think about what we're trying to achieve here. This was the 1.5 degrees report when it came out in 2018. This was a quote from somebody at Greenpeace. Scientists have wanted to write in capital letters, "Act now, idiots." But they needed to say it with facts and numbers. Facts that we're trying to get a fight and flight response out of people. And this is one of the ways where we don't understand human beings. This one says 7 minutes, but go on. We're trying to get a fight or flight out of them. H. sapiens, the miracle actually is that some people really do respond to this abstract explaining of a problem that they cannot see. But they can't fight or flight, they can't fight it, it's global. They can't flee it, it's global. And they can't fight it because it would need a global response that individuals can't do. Most people just ignore it because there's nothing they can do about it. The way to get a fight or flight response out of human beings are biochemical animals. You wave a tiger in front of them, you get adrenaline, you get cortisol, the body reacts, the mind reacts. You explain to them some abstract problem with a report and some graphs. No, it doesn't work. I'll jump past these because Ruth is threatening me. But we are trying to communicate. In the ones that understand it, all it does is create a sense of despair, which is understandable, but it disables us. It makes us less than we are, less capable than they are. This is why conservation optimism was set up. That's a brilliant quote from Figueres. I wish I'd said that, but I didn't. This is what we do. We support, empower, enable sharing hope, building capacity and community. All right. I think I've still got 5 minutes. Framing is everything. Let's use Kahneman's example. The medical procedure that you're about to have has a 10% mortality rate. Try that against the medical procedure you're about to have has a 90% survival rate. They actually engage different parts of the brain. The first one will initiate a fight or flight response. The second one will get the amygdala involved. You get a bunch of serotonin and dopamine. You get happy hormones. Oh, that sounds nice. There is a science to communicating with Homo sapiens in order to get them to shift their behavior. And we need to understand that if that's what we want to achieve. I'll give you a good one. People and nature thrive. How often do we use something similar to that? How often did WWF use it? If we present people and nature as two different things, it's a choice. There is people and there is nature. As soon as the pressure goes on, what are they going to do? They're going to go for people. This is not about people and nature thriving. This is about people thriving. And our biggest challenge is to articulate clearly, I speak to the communicators in the room, articulate clearly the value of nature to people in all its myriad ways. Not, we're doing this for the bears. We may like the bears, but we're doing this for the humans. Why are bears important to humans? In 10 words. That's the challenge. We tell stories not because it's a nice way of communicating, it is the way the human brain works. Somebody once said, we understand the world through narratives and then we go to bed and dream stories. It is the way the human brain works. We tell stories. I'm going to flip past the nice quotes. And we need to get skills. Being a really good scientist and having at some point acquired the power of speech does not make one a trained communicator. There is a real art to this, and most of the time we don't have it, and we need to get it. How many people, and I'm sorry to do this to you, because I like you all, but how many people are going to stick up really complex slides up there with graphs and quotes and charts up, and then expect people to listen to them? H. sapiens vulgaris favours the visual over the auditory. You give them something complicated to look at, and you could read out next week's winning lottery numbers. They're not going to listen to you because they're reading your slide. If you've got a simple slide up there that adds to what you're saying or provides a focus, that's fine. 2.5 minutes. Okay. I can get on to optimism. I'm supposed to be a director of optimism. Breathe. I have got enough time. There is hope here. It's really difficult, and I'm busy telling people, stop worrying about all these animals that you love and how we're going to clean up behind the humans and put them back. That's important. I'm not saying stop it, but we have got to be engaging, and this is zoos I'm talking to. 140 million visitors a year in Europe alone. If we can't engage with them, who can? Your communicators are probably your strongest conservationists right now. But think on this. Despite all the crap and the orange people and the little red hats and everything else, this is the proportion of humans living in absolute poverty. And it's gone down in recent years. That's life expectancy across the world. And even in the poorest parts of the world and the least developed parts of the world yet, it's gone up. This is global child mortality. Under 5 mortality was at 42% for centuries. And now look at it. We can do this stuff. Humans, despite all their foibles, are not that dumb. These are things they value, the life of a child. They can look at people who maybe live in a completely different country and think, those people shouldn't have to live like that, in that poverty. And they push it forward, and they make things better. Simple, fundamental things that humans value. Adding the place we live to that list should not be rocket science. 30 seconds. This is inspiring. I'm trying to remember whether that's my last slide or not. It is inspiring because it means that we don't have to change the world. It means that they will catch up. It's going to take a long time. We're not going to do it by 2030, I think. I hope, but I don't think so. So it's going to take time. But it does mean that they'll get there 3 or 4 generations down the line, 100 years from now, people will have caught up. And they'll look back at us and think, how could they have lived that unsustainably? And it means that every population of butterflies that we protect, every little bit of healthy habitat, every tiny bit of climate change mitigated, totally worth it, because they will catch up, and that little bit will be there waiting for them when they do. Our conservation status is vulnerable and we need behaviour change. Therefore, this is a social science. And I hate to do that to an academic research conference, but it is. We have a huge role to play. And it is going to be a long road, but we'll get there. Work with conservation optimism, work with your communicators. Let's get this done. 3 or 4 years, 3 or 4 generations perhaps, but we'll do it. Thank you. Sorry, Ruth, that was 30 seconds over even by my clock.
Doesn't matter, John. Ruth is coming for you now anyways. Do we have some interesting questions for John? Okay.
Hi.
Hi, thanks. No, that was really inspiring and really nice to hear. And I work a lot in the field of disease ecology. So sometimes these optimistic stories are really challenging to tell. And I just wonder if you could have your perspective on this. So, for example, in zoonotic disease spillover, which is where I work, we kind of know what's going on. The big problem is how we tell people, and we saw the backlash after the COVID pandemic, even though we're pretty certain how it emerged. Yeah. But it's a hard truth to swallow because it means that we need to change our behaviour. And I just, like particularly in that context, how do you reframe that? How do you turn that into an optimistic?
It's not a dissimilar question to the one you asked Marcus earlier.
Yeah.
And when you asked Marcus, I was thinking, that's the rest of the day gone. Yeah, but you asked the fundamental question. This is human behaviour change. Communication is our tool. How do we do it? And if I knew a quick answer to that, I'd give you the first copy of my book. There's a lot of people working on it. Let's sit down over lunch and bash it out a bit. But I can't give you a quick answer because the question you're asking is the fundamental question. How do we get them to worry about spillover? How do we get them to worry about climate change? How do we get them to worry about biodiversity loss? If I knew, I'd be famous, more famous.
Thanks. Yeah, and a chat over lunch would be great because,
Hi. So you talked about our list of, let's say, things we prioritize as biochemical animal. But I'm curious, how do you think our cultural values distort or shuffle up and down those priorities? Like how I think our economic system or our cultural values, for example, value too much or more the economy than the environment, or even human health, or money over other things, and
Two quick thoughts, and then I better get out of the way for whoever's next. One of them is it comes back to articulating the value of nature, and one of the ways nature feeds into us is through our cultural values, and we need to be able to articulate that. And that's going to be different whether you're living alongside tigers in Asia or whether you live in the UK or some other part of the world. It does feed in. But coming back to the nub of your question, I usually think that human beings are fairly predictable. You give me one person in a situation, I will have no idea how they react. But give me 1,000, and I could probably make a fairly predictable guess about how they'll react. But having said that, cultural differences do feed into that, and they will affect those 1,000 people to an extent. And trying to understand that is incredibly important and incredibly difficult. How it influences is going to depend on what culture, under what circumstances, in what particular context. But I don't think it's immune. It's the one thing, I think. Why is it that maybe 1,000 people in Germany might react differently to a similar situation than 1,000 people in Brazil? And that is only the socio-demographic culture change. And we don't understand enough of it. But it is a big factor, definitely.
Thank you.
Good. One last applause for John, and also a nice gift for you also. Yeah, and now we are heading to Africa because we have our next online speaker, Dr. Victoria Montong Yilwa Dan from the Nigerian Defence Academy in Kaduna. Can you hear us? During that pause, I can introduce her. Dr. Dan is an associate professor of genetics and plant breeding at the Department of Biological Science, the Nigerian Defence Academy, and she has carried out researches in various areas of botany, including genetics, plant breeding, herbal medicine, and environment. And the name of her presentation is Combating Challenges of Biodiversity in a developing community. And let's hope that this will work now. Unfortunately, Dr. Victoria Montong is not able to make it live because weather conditions so on, so the connection is not the best. So instead we are playing her presentation, the pre-recorded version of it. I hope this works then, Jan?
Okay.
Yeah. Good. Here's a presentation of Victoria Montong Yilwa Dan.
My name is Victoria Montong Yilwa Dan. I'm from the Nigerian Defence Academy in Nigeria. I'm presenting a paper titled Combating Challenges of Biodiversity Conservation in a Developing Economy. As a way of introduction, biodiversity deals with living things. It provides essential services such as food security, climate regulation, sustainable livelihoods, and cultural practices. Biodiversity is increasingly under threat, particularly in developing countries, because of rapid economic development, population growth, and inadequate governance structures. Developing countries have a significant proportion of the world's biodiversity due to their tropical and subtropical climates, which provide suitable conditions for diverse plant and animal species. This review discusses the significance of biodiversity, conservation challenges, and explores strategies to combat these biodiversity challenges for a sustainable environment. Challenges of biodiversity conservation. These include deforestation, population growth, lack of funding, overexploitation of natural resources, climate change, and weak environmental policies. Deforestation and urbanization. Deforestation occurs due to population growth, urbanization, and industrialization. As land is cleared for housing and road construction, biodiversity-rich areas are also lost. Pollution. Pollution resulting from human activities such as oil spillage and gaseous emissions from industries cause long-term damage to ecosystems. Population growth and resource overexploitation. Population growth increases demands on natural resources, leading to habitat destruction, overharvesting of forests, overfishing, and hunting for wildlife. Poverty and livelihood dependency on natural resources. Limited access to alternative income sources leads to ecologically harmful activities. In developing countries, over 300 million people depend directly on biodiversity for food and income. Illegal logging and mining activities. Illegal logging and mining activities threaten biodiversity by degrading forests and polluting water bodies. Desertification and land degradation. Desertification leads to land degradation, thereby reducing agricultural productivity, displacement of wildlife, habitat loss, and food insecurity. Climate change. Developing countries are more vulnerable to climate change because they depend on climate-sensitive sectors like agriculture. Species can be forced to migrate or face extinction due to climate change. Unsustainable hunting and fishing. Many developing countries rely on biodiversity for food and income, and so tend towards overfishing and overexploitation of wildlife, thus threatening species survival. Illegal wildlife trade. Illegal wildlife trade results in biodiversity loss, particularly in developing countries. It is rampant because of weak law enforcement, corruption, and insufficient funding. Limited community involvement in conservation efforts. In many cases, conservation projects are designed without consulting the people directly affected. This leads to resistance and conflict. Successful biodiversity conservation depends on the participation of local communities. Engaging indigenous and local communities in conservation initiatives can lead to more sustainable outcomes. Poor governance and weak policy implementation. Many developing countries have environmental laws and regulations to protect biodiversity, but weak enforcement limits their effectiveness. Additionally, corruption, lack of transparency in resource management, lack of coordination between government agencies, local communities, and international bodies are also factors. Poor funding and limited investment in conservation programs. Many developing countries struggle with funding of biodiversity conservation and restoration. governments spend more money on activities that harm biodiversity than on restoring or protecting it. Unfortunately, private sector involvement is very negligible. Strategies for combating biodiversity challenges. Some of the strategies involved promote reforestation and afforestation. This can restore degraded ecosystems, provide habitats for wildlife. Tree planting and sustainable forest management should also be promoted. For instance, the Nigerian Defence Academy, under the leadership of the Commandant, Major General J.O. Ochai, has embarked on a 1 million tree planting campaign and have successfully planted over 100,000 species in its environment. Establish protected areas and conservation corridors, increasing the number, size, and adequate funding of protected areas, such as national parks and reserves, which conserve biodiversity. This can safeguard habitats for endangered species. Sustainable economic alternatives. Sustainable economic alternatives to destructive policies, such as illegal logging and hunting, should be promoted. When alternatives like ecotourism is created, wildlife habitats will be protected while creating economic opportunities. Integrating biodiversity conservation into national development plans. Policies that support agroforestry can help conserve biodiversity while improving livelihoods. Government should enhance capacity of its agencies for effective implementation and enforcement. This will promote transparency and accountability in resource management and integrate biodiversity conservation into national development plans. Implementing and enforcing conservation laws. There is need for strengthening and implementing strict environmental policies to address current challenges, as well as regulate activities that threaten biodiversity. This will ensure that perpetrators of illegal activities such as poaching, logging, and habitat destruction are held accountable. Using advanced technologies for monitoring biodiversity. Technologies such as satellite imagery, remote sensing, and geographic information systems can be employed to help track changes in land use, detect threats to biodiversity, and monitor deforestation, habitat loss, and illegal activities in protected areas. Public awareness and education. Raising public awareness about the importance of biodiversity conservation is vital. Integrating biodiversity conservation into school curricula can foster environmental responsibility among young people. Conservation clubs and field trips to national parks and reserves can also inspire students to protect biodiversity. Climate adaptation and sustainable development. Promoting agroecological approaches and adopting climate-resilient agricultural practices which integrate biodiversity into agricultural systems can also enhance resilience and sustainability. These practices include planting drought-resistant crops and adopting water-efficient irrigation techniques. Additionally, the use of native species in land restoration should be given priority. Promoting community-based conservation and indigenous knowledge. Local communities can be useful in biodiversity conservation, particularly in areas where they depend on natural resources for survival. Recognizing and integrating indigenous knowledge into conservation strategies can improve biodiversity protection. Community-based conservation programs give local people ownership and incentives to protect wildlife and habitats. Engaging media and social platforms for conservation advocacy. Media campaigns and social media platforms can raise awareness about biodiversity issues and encourage public participation in conservation efforts. Documentaries, community radio programs, and online campaigns can highlight the importance of biodiversity and mobilize support for conservation initiatives. Combating illegal wildlife trade. Strengthen law enforcement to combat poaching and illegal wildlife trade. International cooperation should be enhanced to address cross-border wildlife crimes. Additionally, raising public awareness about the impacts of illegal wildlife trade will significantly improve biodiversity protection. Enhancing international cooperation and funding. Developing countries often lack sufficient funds for biodiversity conservation. However, if national biodiversity goals are aligned with international standards, developing countries can secure funding, technical assistance, and knowledge to address their biodiversity challenges. In conclusion, the conservation of biodiversity in developing countries is crucial for environmental sustainability. While challenges threaten biodiversity, opportunities and strategies that can improve conservation efforts also abound. Adopting the measures outlined in this paper will assist developing countries safeguard their rich biodiversity, not only for them but also for the global community, as biodiversity loss poses a significant threat to the planet's ecological integrity. These are some of the references consulted in the preparation of this paper. Thank you for listening.
Thank you very much, Victoria, for this presentation. If you have any questions here or for the online people, sure, we could collect them and send it to Victoria afterwards. Yeah, now we have Pavel Borecki from the zoo in Breslau. And he's been working at the zoo in Breslau for 15 years, specializing in the care of marine animals, including pinnipeds and African penguins, while also leading the department. And he's currently pursuing a PhD focusing on creating a biobank of genetic resources for critically endangered African penguins. So the name of your presentation is Building a Biobank, a one-plan approach to African penguin conservation. Yeah, the stage should be yours. Is the presentation ready? Good.
Hello, everyone. Sorry for my voice, but I'm coming down with something. Yes, let's just jump into it so you don't have to wait for your lunch break. So I will tell you about our project of building a biobank and how are we attempting to follow the one-plan approach to species conservation with that. A couple words, why are we focusing on African penguins? It's only one native species of penguins from Africa, and among 18 other penguin species, it's one of the most endangered. Last year, it has been uplifted to critically endangered by IUCN. So in the beginning of 19th century, there were over 3 million individuals, and now their wild population declined to less than 20,000. And the reasons for that dramatic decline, it's obviously mostly human-made. So it started with guano scraping, which has destroyed their nesting habitats, and also extensive egg collection was one of the reasons benefiting that terrible decline. And nowadays, the environmental disasters such as oil spills are causing the decline as well. And obviously, the penguins cannot compete with commercial fisheries. So what is African Penguin EEP doing currently? So we are basically keeping the stable, genetically diverse captive population. We are obviously educating people about penguins. And we are we are supporting and fundraising for in-situ conservation projects such as SANCOP, which they do the rescue, rehabilitation, and release programs. But what we also wanted to incorporate into this strategy is building a biobank. That is why 2 years ago I have started my PhD program within our University of Environmental and Life Sciences in Wrocław. And it is financed by our Ministry of Higher Education. So this project, with this short abstract, obviously there are the reasons for decline, and in our project we are trying to focus on coming up with a semen collection method that we are assessing the semen quality, and then we will come up with the cryopreservation of the genetic diversity. And at the end, since the project is the implementation project, we're going to create a guidebook on how step-by-step to create a biobank within a zoo or other facility, and it will be shared with all of the stakeholders that would be interested, for example, IAZA or other organizations that care for penguins. So for our project, we had to choose some specific individuals to take part in this project, and we set set of criteria. So we chose the males depending on their age, overall health and conditions, as well as the rearing method and their pedigree, since there have been cases of hybridization with Humboldt penguins within the European population. And we also focus on whether they had partners and their overall breeding parameters. Then it comes to collecting the semen samples, and that's how we do it. So we've basically applied the dorsal-abdominal message technique that was developed by Barrow and Quince way back in, I think, 60s or 70s. And basically, it means that one person needs to restrain the penguin, covering its head to protect it from biting. Then the second person is performing the dorsal-abdominal message, which is basically stroking the back and the underbelly of a penguin. And afterwards, we are gently squeezing the sides of the cloaca. That makes the cloacal folds to evert. And with a proper physiological reaction of the bird, the semen appears on the cloacal folds, and we can collect it with the capillary. So then with the collected samples, we run some analysis using the smears, computer-assisted sperm analysis, and also with flow cytometry. And we were able to publish some of the papers. So the first one that we published was on general breeding behaviors. And one of the most important conclusions, in my opinion, is that when you feed the captive population or the population in general ad libitum and provide them with a proper environment, they basically can breed like bunnies. So that is an important indicator for species conservation, because it highlights the need to establish the no-take zones in the wild population. And in the second paper, we focus on semen collection and semen evaluation. And from my perspective, I think the most important conclusion is that we are able, with our semen collection method, to collect samples from both habituated and unhabituated individuals, which indicates that it could also be done with wild individuals. Then we moved to cryopreservation as a conservation strategy, and we came up with our own cryopreservation protocol, which you can see here in a short. We are obviously using liquid nitrogen, and then the semen samples that were previously equilibrated with cryoprotectants, then they are moved into those plastic straws. Then the straws are being sealed with the special sealer. Then they are cooled down in the water bath. And afterwards, we are placing them on the rack above the liquid nitrogen in the vapors of it. And afterwards, they are plunged into the liquid nitrogen itself, then moved to the plastic tube, secured. And like that, they are moved to the liquid nitrogen containers. And in that state, they could be kept indefinitely. Sorry. Now going back to the one-plan approach, it has been developed by IUCN, and it's a conservation strategy that is basically promoting holistic and collaborative effort of both in situ and ex situ conservation efforts. It brings together aquaria, zoos, local communities, governments, and conservation organizations, but also scientific institutions, to kind of bridge the gap between in situ and ex situ conservation efforts. And it's obviously been promoted by both IAZA and WAZA. And I would like to show you how we are attempting to do it. within our project. So back in Poland, we've obviously keep our African penguins at our zoo, and we are collaborating in our project with the university, and the finance are coming from the ministry. And on a European level, we are obviously collaborating with the entire IAZA, and are endorsed by our EEP coordinator and IAZA Biobank coordinator as well. And then in South Africa, we are working with the University of the Western Cape, but also with the conservation organizations such as SANCOP or BirdLife South Africa. We are also working with rangers from those nature conservation groups, and also with a variety of zoos and aquaria, with one collaborative goal to save this endangered species. And then I think the most important thing, in my opinion, in this one plan approach is actually talking to people, because you cannot get everyone on board if you cannot talk to them. I think that was also the thing that yesterday's presentation was also about. It's very hard to connect with people. So I was able to go to South Africa in the beginning of this month. This year, I spent a month over there, connecting in person with all of those people that I'm presenting here, because you can send a lot of emails, but you will not get a response. And then when you go there, as it was also said during the last evening's presentation, a lot of those institutions are competing for the funds. So it's very hard to get the common ground with them. But if you actually go there and talk to them, it's more doable if you're actually talking to a real person, not via email. So that is all for me. So thank you for your attention, and if you have any question, I'll be glad to answer.
Yo. Thank you, Pavel, for this beautiful presentation. Do we have any open questions here? Okay, Paul. Okay.
Thanks for your talk, really interesting. Could you just expand a bit on the ad lib feeding? You mentioned that was something really important for captive breeding.
Yeah.
How do you do that in practice, and what is it like for the zookeepers' feeding regime? How do they know how much to give?
So basically, well, I've been working with those African penguins for the past 10 years, so I've been their keeper as well. So what we do, because we have one of the largest colonies in the world with over 100 individuals, so you cannot feed them individually. You would have to have at least 10 people in the team to keep score. So we feed 2 times on the beach, and whoever wants to come, they come to the bucket and feed. But we also do 2 feeds in the pool. So we just give them as much food as they like. So that's the ad libitum. Like the IAZA African penguin manual says that African penguin could eat around ½ kilo of fish a day. So we prepare more or less that for entire day, and we just give them that. If we see that they're not hungry and, for example, the fish is starting to fall down to the bottom, we stop the feed. But then if we see that they, for many reasons, want to eat more, we'll just give them more.
Thank you.
Mm-hmm.
Thank you very much. More questions? No. Okay. Then thank you very much for your presentation. For the online attendees, don't forget that Pavel is also available for the best talk presentation. So after this session, you can vote for it. Also for like the usual presentation which we had already.